Okay, should we start now? Okay, so let's start. Good morning. It's my great pleasure to introduce Dr. Liu. Rujuan is an assistant professor at Shanghai Tech University since 2019. She got her PhD as a joint program of the University of Kentucky and the University of Science and Technology of China and the Chinese Academy of Science. She was appointed by the Chinese Academy of Science as the associate professor and got promoted as the professor. She is also the visiting scientist of EMBO Grenoble Outstation in France and the University of Chicago. Her lab focuses on biogenesis and function mechanism of human disease-associated tRNA modifications in mammalian system using human cell lines and mouse models. So her talking title today is the Catalyzing tRNA, mRNA, or others, the multifaceted role of tRNA modifying enzymes. So let's welcome her.
Thank you, Zhao, for your introduction. And good evening, friends from the East, and good afternoon or good morning, friends from the West. And I'm starting sharing the screen. Okay. Paul, can you see my slide?
Yeah. So you just need to go to the first slide and play the,
Okay, now.
Yeah.
Okay, thanks, Paul, for your introduction. And I'm very glad to have the opportunity to share some of our recent work progress with you here. And today I'm going to talk about the multiple function of tRNA modifying enzymes. And we all know that now there's more than 170 different types of modifications on the RNAs, and many of them are closely related with human disease, all the red ones here. And we know that high throughput sequencing has been developed for some of the modifications, but unfortunately only very few limited ones, the green ones. And the last 10 years, modifications on mRNA, especially the M6A, has been extensively studied, and it has been shown to be a very important modification to involved in every aspect of cell processes. And indeed, the RNA modifications is also heavily modified on other RNAs, on other non-coding RNAs, for example, tRNA, ribosome RNA, small nuclei, and other RNAs. Indeed, of all the cellular RNAs, tRNA is the one with the heaviest modification. And when comparing with mRNA, we can see that in mRNA, there's less than 10 modification types has been identified. But for tRNA, it's already more than 100. And the ratio for mRNA, the average modification ratio is about 0.1%. However, the ratio for the tRNA could be reached as high as 20% for some tRNAs. And now the more than 100 different types of modifications has been identified on tRNA, which have a classic clover-leaf secondary structure and the inverted L-shaped tertiary structure. And those modifications has been shown to involve in regulating the tRNA functions, including the classical function that the tRNA working as a classical adapter molecule between mRNA and amino acids. Beyond protein synthesis, tRNA also have many other functions. For example, tRNA itself has been working as a regulatory RNAs, and And TI is also the precursor of many regulatory TIs, TI fragments, and TI-derived small i's. And TI itself is also working as primers in some retrovirus. So which means that the modification could affect both the protein synthesis and all the non-kinetical functions of TI. Because of and actually TI modifications has also been found to be closely associated with human disease, because many mutations on the putative TI modifying enzymes has been identified with 3 main types of human disease, which is the neurological disease, cancers, and metabolic disorder. So my lab is focused on the biogenesis and the function of those disease-related TI modifications. We're working on the writer, eraser, and the reader to understand its biogenesis and function, mainly of the 3 types of modifications in my lab: the NM modification, M2G, M22G, and the M5C one. And today I'm going to focus on the writer, the TI modifying enzymes, the so-called writer. And first I will briefly go through the modifying enzymes we identified or characterized recently, which is the NM modification at the acceptor stem or at the anticodon loop region, or the M5C and the M2G at the acceptor stem or the anticodon loop. And we find that many of the TI modification enzymes, modifying enzymes, they could working as a single protein, which means that they can perform the TI modification stand alone independently by itself. But many others need to work together with the auxiliary proteins. For example, the T112 or the WDR6. They are all auxiliary proteins working together with the master transferase to perform TI modifications. And we also find that some of the TI modifications, especially those ones in the anticodon stem loop of TI, they have a very close crosstalk between each other, which means that some of the modifications are interdependent on each other. For example, in E. coli TI, we find that the I6A modification at site 77 is essential for the NM modification at site 34. And in human TI filarin, we also find the M1G 37 is essential for the GM34. And in the human TI valine, we find that the inosine modification at site 34 is essential for the modification at the site 78. And also through our study of many of these enzymes, we found out that many of the TI modifications are dependent on its tertiary structure, the L-shaped tertiary structure. For example, the enzyme 6, which catalyze the M5C modification here, and also the TRIMJ, which catalyze the NM72 here, and also others. And through a structural study, we find out that for the TI modifying enzymes that recognize the TI's 3D structure is mainly fulfilled through recognizing multiple elements of the TIs. Taking the enzyme 6, for example, resolved its complex structure with the TI, and we find out that the characteristic 3' CCAN of TI is recognized, and also the specific sequence at the acceptor region of the TI is also recognized by the modifying enzymes. And also the D arm here is also recognized by the protein. So through multiple elements, the TI modifying enzymes could specifically catalyze its own substrate TIs. However, we also find that some of the TI modifying enzymes, they do not have such a specificity dependent on the tRNA tetrary structure. Instead, it could catalyze just a single stem loop structure. For example, the trim L, we identified that it could using a stem loop as its substrate. And for this kind of modifying enzymes, its specificity toward the tRNA is remaining to be determined. And actually, during our study, we found that many of the tRNA modifying enzymes have an extensive binding with other RNAs, including its substrate tRNA, non-substrate tRNA, and also many other RNAs, including mRNA and some linker RNAs. Actually, we try to identify whether they are non-specific binding or specific binding. Unexpectedly, we find many of the binding are quite specific. Actually, the many CRISPR data identified by other groups on other enzymes also reveal that the tRNA modifying enzymes could bind many other tRNAs beyond, could bind many other RNAs beyond the tRNA. And actually, recently, through the high throughput application for identify RNA modifications, for example, the PSI and the M5C and the M1A modifications, many scientists identify that the modifications on mRNA are indeed performed by the tRNA modifying enzymes. But as we just showed that some of the tRNA modification enzymes, for example, the enzyme 6, which have a very high specificity towards tRNA, how it be able to catalyze mRNA? Do they need some auxiliary proteins? I mean, do they need some accompanying proteins or chaperone proteins to help them to recognize the substrate or the other mechanism? And then we try to identify many, we try to characterize many of those tRNA modifying enzymes on how they work with the mRNA. And to our surprise, we found that the tRNA modifying enzymes could efficiently catalyze mRNA and some non-coding RNAs as efficiently as tRNA. And for some of the enzymes, its modification activity toward mRNA is even higher than that for the tRNA. So which means that the activity is truly in vivo, and it's not just the remain activity from the tRNA. So the next 15 minutes, I'm going to focus on one of our recent work, which mainly done by postdoc students in my lab, Li Hao. It's about a human tRNA modifying enzymes, which have a dual role in both regulating translation and transcription. And this enzyme is catalyzing the NM4 modification, which is a modification that adding an methyl group as the ribones of the nucleic acid. And in mammals, the tRNA have many sites of the NM modifications, and which are performed by different tRNA modification enzymes or complex. For the site 4, its modifying enzymes in mammals has been remain unknown until this work. And in yeast, is the TRIM13. The TRIM13 has been identified as the master transferase. So in mammals, so in human, we first identify the protein named the co-echoid containing protein 76, which have 2 zinc fingers and 1 co-echoid domain and a master transferase domain. And we identify that this protein is the human homolog of the yeast TRIM13. And when we knock out of this gene, we find that the NM modification is totally diminished in some of the tRNAs, for example, tRNA glycine CC and the glycine proline. And on some of the other tRNAs, the NM modification level dropped a lot. And further, we purified this protein and performed the in vitro activity towards towards some TIs, and we found that it could be highly efficiently methylate TI glycine and TI proline, but not TI histidine. And its enzymatic activity was further checked by mass spec. And further, we show that the modification is indeed happened at the site at the position 4 through replacing the site 4 from U to the ACG, and we can see that when the NM modification, the AM, CM, and GM could be identified when the mutation happened. So this simple reaction could tell us that the CCDC76 is indeed the NM4 modifying enzyme. So we rename it as the TRMT13 as what in the East system. And further, we show that this protein recognizes the tertiary structure of TI, which means that it cannot catalyze the stem loop structure. And further, we performed the iClip for the TRIM13, and we identified that TI is its main substrate. But we also observed MI and some other link Is. So what's the biological function of this protein? And firstly, we find that this protein is highly expressed in breast cancer cell lines. And in the breast cancer cell lines, and through analyzing the TCGA database, we find out this protein has a strong correlation with the EIF, which is the translation initiation factor. So then we performed the polysome profiling and the nascent peptide synthesis, and we identified that the knockdown of the TRIM17 expression could decrease the global could decrease the global protein translation in vivo. And interestingly, we find this activity is dependent on its TI modification, because when we mutate its catalytic essential residue, the E463, we can see that mutation could not rescue the knockdown of the TRIM17. And further, we show that the loss of this modification will not affect the steady level of the substrate TIs or the amino acylation level, which means the charging level of the TIs. Further, we identify that the TRIM13 modulates protein translation mainly through modulating the production of the TI-derived fragments. The TI-derived fragments could modulate the protein translation, as firstly reported in 2011 by Paul Anderson's lab in Harvard. And we identified that the TRIM13 depletion could increase the level of the one of the TI-derived fragment, the TI glycine CCC. And we can see that the fragment could dramatically inhibit the protein the nascent peptide synthesis, and the full-length TI glycine will not. And interestingly, although we see the protein synthesis defects of the cell, but there's no obvious defect on the cell proliferation upon the knockdown of the TRIM17. And further, we found out that the TRIM17 itself could promote breast cancer cell migration. We can see that when we knock down these genes, when we knock down the expression of these genes in the breast cancer cell lines, the cell migration is largely inhibited. And this, interestingly, this activity is independent of TI modification, because when we are using the mutant, we can see it could rescue the cell migration as well as the wild type ones. So which means that the TRIM17 TRIM13 promote cell migration is independent of the TI modification activity. And further, we show that the EMT pathway, and the many cell migration and cell junction pathway is affected when knockdown of the TRIM7, when the knockdown of the TRIM13, and we can see that all the EMT markers is decreased. And further, we try to understand its mechanism. Interestingly, we find that the TI is co-localized with the TRIM13 only in cytoplasmic, but not the nuclear ones. But we do see the clear localization of the TRIM13 in nuclear. So what's its function? Whether it's bind with some other TI, other ions beyond TI. So we performed the RNA-IP, and after sequencing, unexpectedly, we find that in all the TI is identical, all the TI is binding to the TRIM13 is only identified in cytosol, but in the nucleus we can see the larger molecular weight nucleic acids could be pulled down with the TRIM13, which means that it could be binding to other nucleic acids, but not TI in the nucleus. And further, we show that the TRIM13 have a co-localization with the chromatin. And actually, we further performed ChIP-seq, and we find that TRIM13 is indeed bound to the promoter region of the genes, and it have its binding motif is as this. And when we analyze the motif with the other transcription factors, surprisingly, we found that TRIM13 is indeed like a promoter, like a transcription factor or co-activator. And we can see that the binding motif of the TRIM13 is very similar to some of the famous transcription factors, including FORCE and JUNB. And further, we've identified that the TRIM13 in nuclear is indeed directly binding to DNA, but not single-strand RNA. And we performed the in vitro EMASA assay using the purified the DI, and we can see this modification. We can clearly see the shifted DNA bound band here. And further, we find that the TRIM13 itself could working as a transcriptional activator to promote the transcription of the TGF-beta, PKN2, and MAF2A. So which means that the TRIM13 could promote transcription independent of its TI modification activity. And further, we identify that the TRIM13 and other known transcription factors indeed have a co-localization and the interaction in vivo through the IP study and also the dual-link study. And we further find that the TRIM13 itself could promote the transcription could promote the transcriptional modulation activity of those known transcription factors. And further, we find that the TRIM13 bound to TI or DNA are exclusively because when the TRIM13 bind with the transcription factors, it will kick off TI, but binding strongly with the double-strand DNA, as the biochemistry studies here. And further, in the mouse study, we find that the knockdown of these genes could significantly attenuate breast cancer lung metastasis. And for the patient samples, we also find that the TRIM13 gene is highly expressed in the breast cancer cells, especially in those samples with the worst survival. And through the bioinformatics study, we can find that the TRIM13 is indeed have a correlation with the TGF-beta1, PKN2, MAF2A, which those genes are known genes that could promote breast cancer metastasis. And we can see, and actually, as we identify, they are the downstream of the TRIM13. So which means that the TRIM13's transcriptional function to modulate the downstream transcription factors is the main cause of the breast cancer, is the main cause of the TRIM13 induced cell migration in the breast cancers. So for our summary, we identified a TI modifying enzyme TRIM13, which have a dual function in both the translation and the transcription. Its translation activity is dependent on its TI modification activity, while its transcription activity is independent of enzymatic activity. Indeed, it's dependent on its DNA binding activity. So for summarization, as I have mentioned before, that many mutations as the TI modifying enzymes has been identified to cause human disease. And now we know that the human TI modifying enzymes could both have the catalytic function and also non-catalytic function. The catalytic function, it could have substrates as TI, MI, linker, and others. And for its non-catalytic function itself, we know it's a RBP, it's a binding protein. So it could working as a nucleic acid binding function, and it is also just a protein. So we know that to understand its mechanism of how the mutation lead to human disease, we should take all these functions of the TI modifying enzymes to understand its final mechanism. And lastly, I would like to thank all the lab members in my lab, and also thank all my collaborators and all the fundings. Thanks all. I would like to take, thank you all for your attention, and I would like to take questions. Thanks.
Thank you, Rujuan. So that's a very interesting, very nice study. You are welcome to put your question on the Q&A part. We don't really have enough time for every question, but we will, Rujuan can type her answers for your question. I will just ask very short questions. So what makes the TRIM13 bind to the RNA or the transcription factor? Is just because of the concentration of the tRNA or the concentration of the transcription factor, so make a switch between those two partners?
Paul, thank you very much for your question. And I think you are asking a very important point of the TRIM13. Indeed, till now we do not know why the TRIM13 how it shuffles between the cytoplasma and the nucleus. And we try to identify whether there are any how to say, for example, the environmental change or some stress could cause the enzyme to move from the nucleus to cytoplasma or the vice versa. And indeed, when we deprive the FBS in the medium, we do find that the cytoplasma, the protein of the TRIM13 is almost disappear, but the nuclear ones is accumulate. So I guess there's some dynamic between there's some dynamic in the TRIM13 in vivo, but now we guess there should be some post-translational modifications, for example, like phosphorylation or others, but we still do not have the final answer yet.
Okay, thank you. So for the time limitations, we will move to our next speaker, Hui. So you can still put your questions on the Q&A part.
Okay,
so,
thanks.
Thank you. So Hui received her bachelor from Nanjing University in 2008, and then received her PhD in biochemistry and molecular biology from Shanghai Institute of Biochemistry and Cell Biology in 2014. After that, she joined Dr. Ding Xiao Patel's lab in Memorial Sloan Kettering Cancer Center. Her research interest is on the mechanistic insights into prokaryotic adaptive CRISPR-Cas immune systems, including precise recognition and cleavage mechanisms and the inhibition of these effector complexes. Her talk title today is Structural Insights into Substrate Targeting and Inhibition of CRISPR-Cas Systems. So let's welcome Hui.
Thank you.
So you change how to change the spotlight into Hui.
Can you see? Does it work?
Now I cannot move to the screen to Hui. How do I do that? Barbara, can you help? Oh yeah. Okay.
It works?
Yeah, it works.
Oh, thank you. Thank you for the introduction, and thank you for the invitation. And thank you for everyone for attending this seminar. I'm Hui. So today I'm going to talk about something about CRISPR-Cas systems. The topic is a large topic. I'm going to narrow it down later. So when we talk about CRISPR-Cas, everyone may think about the Nobel Prize in 2020. The two female scientists win the Nobel Prize in chemistry for their CRISPR-Cas tools, because these two scientists, they developed a way to edit genes by CRISPR-Cas systems. It works like molecular scissors, and it's widely used in several ranges. So actually, why they can win the Nobel Prize? Because the CRISPR-Cas systems is widely used in not only DNA but also RNA manipulations. It can actually knock down the DNA sequence, introduce indels, or introduce some genes that you want to insert in the particular site in the DNA, or it can knock down RNA. Also, it can do single base editings both in DNA or in RNA. Also, when it binds to some fluorescent proteins, it can do tracking and imaging in lab cells for DNA or RNA. Also, when it linked to some other components like activators or repressors, it can change the modifications in DNA or RNA. So these powerful tools based on CRISPR-Cas systems, why it can target such both different nucleotide gases. When we look back to these CRISPR-Cas systems, actually it's adaptive immune systems in prokaryotic cells. So when here I show a typical CRISPR-Cas gene locus, it contains a lot of genes encode related Cas proteins. And also it contains a CRISPR array, which contains several sequences we call the repeat, which is the same sequence in a special species. Then each two repeats is separated by a sequence we call the spacer, which is different in the CRISPR array. Once the phage infect the, like we say, a bacteria, the injected fragment of viral DNA can be introduced and inserted into the first repeat and form a new spacer. This step we call acquisition, which is similar like antibodies in human beings. It can memorize the invading fragments of DNA. Then the CRISPR array can transcribe into a precursor CRISPR RNA, which contains several different spaces, as I shown here. Later, it will be processed into individual mature CRISPR RNA. These steps can be finished by either the host genes RNA or by the Cas proteins in different systems. Then later, the mature CRISPR RNA and the Cas proteins form complex, ribonucleotide complex, we call the effectors. Here we've got two different effectors. One contains only single Cas proteins and a single single CRISPR-RNA. The other is contains multiple Cas proteins and a single CRISPR-RNA. So it can be divided into two systems. The multiple Cas proteins we call the class 1 CRISPR-Cas systems, and the other one contains only single Cas proteins we call the class 2 systems. Both of these two systems can target sequences specifically target both DNA or RNA. So generally speaking, the basic mechanisms of CRISPR-Cas systems to target DNA or RNA, it follows some rules. So first, the Cas proteins and the natural CRISPR form a effector proteins as shown in the last slide. Then is if we go to the way to recognize DNA, it should be initialized by a PAM scanning. So once it finds a PAM sequence in a double-strand DNA, the Cas RNA proteins can bond with this special DNA, and then it will melt the DNA double-strand break. Once one strand is paired with the spacers, the DNA can be either cut by the Cas proteins, or it can be transported by some T7-like transposons. So when it recognizes RNA, as I shown here, the single strand RNA, once it pairs with the spacer regions, it will be digested by the Cas proteins. During the battles between CRISPR-Cas systems and the phages, the phage wants to fight back the CRISPR-Cas system. So it invoked a lot of anti-CRISPR proteins. First, these anti-CRISPR proteins can either block the cleavage of DNA, or it can block the recognition or the cleavage of DNA, or it can block the cleavage of RNA. Once it blocks the activity of the Cas RNA ribonucleotide complexes, the phage can escape from the CRISPR-Cas immune systems. So actually, when we use the Cas tools, this kind of anti-CRISPR proteins can work as some kind of switches to turn on or turn off the tools. Here, I'm going to talk about two stories. One is about the RNA-guided DNA integration, and the other one is how to generate the RNA cleavage here. So first, when we look into the DNA cleavage, and we're going to talk about class 1 effectors today, which I mentioned before. The class 1 effectors contains multiple Cas proteins and a single CRISPR RNA. So we talk about today is type 1 CRISPR-Cas systems. It can subdivide it into several subtypes. So we focus on here, as I showed, type 1F. Here, the Cas1, Cas2 proteins is responsible for acquisition, which I mentioned before, which it can integrate the invading DNA into the CRISPR array here. Then the Cas6 proteins is responsible for precursor processing. Here, the Cas6, Cas5, Cas7, and Cas8, the 4 subunits. Here, the 4 types of subunits can, together with the CRISPR-RNA, can form multiple Cas proteins with complex we call the Cascade or CSY. This complex will recognize the double-strand DNA. Once it recognizes DNA and melt the double-strand DNA, it will recruit the Cas3 subunits for the cleavage of both DNA strands. So before we start today's proteins, we did some works before I came back to China. Here we determined the Cascade or CSY proteins with protein complex with DNA. Let me remind you first, when we bind this double-strand DNA, there's 2 factors that is very important. First is PAM sequence. It will initialize the recognition. And second, it should contain a sequence which can pair with the spacer regions. Here, the cartoon models of the CSY complex, also we call the Cascade complex. It forms by several subunits. First, the 6 copies of Cas7 form a backbone of the whole complex. Then the Cas6 proteins, which is responsible for CRISPR-RNA processing, it will bind with the helping region with the CRISPR-RNA, and we call this region as a head region of this whole complex. And the Cas8 and Cas5 complex, they form a heterodimer and located at the tail of the whole complex. It will be responsible for the recognition of PAM sequence here. Here is the overall structure. It's formed like a G-shape whole complex. I can show you. Okay, here is the whole complex as a spring or like a G. So here is the backbone of the whole complex. The one side we see a Cas6, and the other side we can see the Cas5 and Cas8. Okay. So there are some type 1F variants which is different with what I talk about, because in this sequence they can only the Cas3 genes is missing in this sequence, meaning the Cascade complex cannot cut the double-strand DNA. So how can it protect the bacterial cells? Scientifically, they found the Cas gene locus is co-localized with TN7 transposon gene locus. So the transposon contains several subunits. We call the TNS A, B, C, and D and E. The TNS A and B is responsible for the transposon. They are transporters. And TNS C is regulates the catalytic activity of TNS A and B. And TNS E and D is responsible for the sequence recognition for the transposons. When it co-localized with Cascade, people thinking, could it be possible that TNS7 transposons work together with the Cascade complex? So in 2019, a group they found that once they the Cascade and the TN7 transposons can guide the sequence, the special DNA cargoes into the special sequence, as I show here. A knockdown of each of the subunits will block the DNA integration into a specific size. And the transposon, they can introduce the different size of the DNA, which means it has a large advantage than the traditional knock-in technologies. Because when we use Cas9 and Cas12 for knock-in, it relies on the HR or NHEJ for the knock-in sequence, but it limited by the length of the sequence. If you use a transposon, the size of the DNA can be largely into around 4 kilobytes. So it is a brand new and very useful technology for future. So they propose possible mechanisms here. The Cascade may first it will recognize the double-strand DNA, then it will recruit the TN7 transposon subunits tNq and here TNSC. Then they will recruit the TNSA and B to insert the DNA sequence into the low downstreams of the Cascade recognized sequence, as I show here. So what's the mechanism here? We actually determine we reconstruct this whole complex in vitro, and we determine the cryo-EM structures of the Cascade together with the tNq. And we found the tNq from a homodimer and located at the head of the Cascade complex. This complex is quite similar as we reported before, and the Cas7 also formed the backbone of the whole complex. We checked each subunit in this new Cascade sequence systems, and we found each subunit from resembles the similar structures as we report before. Then we look into the interface between Cascade and the tNq. So we found the two homodimer interact with the Cas6 and the Cas7 individually. So we show the detailed interaction here. The interactions involved a lot of hydrogen bonds and salt bridge here. We introduced mutants at the interface, and we found once we block the interactions between the tNQ and the cascade, it cannot put down the mutant tNQ cannot put down the cascade complex. Then we try to reconsume this whole complex in vivo to see whether we can achieve the in vivo DNA integrations. As I show here, we use a 1 kilobit cargos gene as insertions, and when we use wild type systems, we can see the bands of the insertions. And if we introduce the mutations which block the interaction between tNQ and the cascade, we cannot find the DNA integrations, meaning the interactions between tNQ and the cascade is essential for the in vivo DNA integrations. Then when we look into the tNQ, it forms a head-to-tail homodimer, and we found there's 2 zinc fingers here, which is important for the whole structures of the tNQ. Then we look into the dimerization interface between those 2 tNQ monomers, and we found it stabilized by several salt bridge and hydrogen bonds. Similarly, we introduce mutations at these regions to block the dimerization of tNQ. Then we found these mutations will reduce the interactions between tNQ and the cascade. Similarly, if these mutations will block the in vivo DNA integrations. Then we will start to thinking what the situations if whether it can recognize DNA in the same way. So we determine the cranium structures of a cascade of tNQ when it bonds to double-strand DNA. Here, as I showed, the Cas5 and Cas6, Cas8 subunits recognize the PAM region from the 9 group of the DNA, and also the Cas7 pin the whole CRISPR-RNA into the whole complex and wing it by the beta hairpin in each subunit. Once it binds with DNA, we can see it forms a hetero RNA-DNA duplex, and each duplex was stabilized by the hairpin with the Cas7 beta strand hairpin. Then we compared the structures before the DNA loading and after DNA loading. We found here shows is the CRISPR-RNAs. We found there's minor domain movements between these 2 structures, meaning the DNA loading induced minor conformational changes. Okay, here's the take-home messages. We found that tNQ exists as a homodimer, and this homodimer is essential for the in vitro and the in vivo DNA integrations. And tNQ interact with the Cas6 and the Cas7 in the head region of cascade. And blockage of the recognition between tNQ and the cascade will reduce the in vivo DNA integrations. Also, the PAM is recognized by Cas8 subunits from a minor group, and the minor conformational change will induce upon the DNA loading. Okay, I'm going to talk about the second project, which is about the RNA targeting and cleavage. This time we talk about the single Cas proteins. Cas13 is the only one sequence found to target DNA by a single Cas proteins. It can subdivide into several subtypes, and in each type, the composition of the whole gene locus and the length of the Cas proteins and also the structures may have divergent, but the common feature is the happen domain. It exists in all Cas13 proteins. The common features for the Cas13s to target a single strand RNA is when it bound with CRISPR-RNA, the Cas13 exists in active inactivated state. Once it bonds with target DNA, it was activated for both RNA cleavage activities. First, it will cleavage the target RNA. We call it a cis cleavage activity. And once it bonds with the target RNA, it will cleave the environmental single-strand RNA in a non-sequence specific manner. We call it trans cleavage. So based on these two cleavage activities, they can be used in several fields. First, it can be used for sequence-specific RNA knockdown, or the RNA-based editing and imaging, and also RNA modification. And useful trans cleavage activities, it can be used for nucleotide acid detections. One is bound with the target RNA, it will cleave with the RNA monomer. The cleavage of the RNA monomer will release the fluorescent signals. Today we're going to talk about the Cas13a subtypes. Actually, before ours, there's a lot of structure and biochemistry studies for the Cas13a systems, and they exclude the apiform, pre-target bound, and also target bound structures, and they exclude the pre-CRISPR processing and also the target RNA binding mechanisms in these Cas13a systems. The key question is, how could these Cas13a systems distinguish the self and the non-self RNA? As in DNA systems, they use PAM, but in Cas13 systems, they don't use PAM systems. They use tag systems, as I will show here. The tag systems actually is a special sequence in CRISPR RNA, which is adjacent in spacer. Once the target RNA is paired with the tag region, we call it anti-tag. It will block the cleavage activity here. Also, when we check the cis cleavages, when the CRISPR is not paired with the tag region, we can see it will trans cleavage the environmental RNA. And when the RNA contains the anti-tag sequence and form a duplex with tag region, then it will block the cleavage activities. So here. Also in vivo activities, the anti-tag region paired with the tag region of the CRISPR RNA will block the in vivo RNA residence for bacteria. So the proposed mechanism is here. When the target RNA contains an anti-tag region paired with the CRISPR RNA, then it will block both cis and trans cleavage activity of the Cas13. So first, we look into the different species in Cas13a systems. Here we highlight 3 different species, and we found the CRISPR RNA tag region identical in these 3 different species. So we are thinking whether they have the similar discrimination mechanisms. So we designed 3 different RNAs. RNA1 contains no parent region, and RNA2 and RNA3 contains different lengths of anti-tag regions. We found that when the target RNA contains the anti-tag regions, they will block the cleavage activities of the both cis and trans cleavage activity of Cas13. And then we ask whether these cleavage activities will influence the in vivo RNA knockdown. We check these 2 different lengths of anti-tag in vivo, and we found that if we introduce different lengths of the anti-tag, both of these 2 lengths of anti-tag will block the in vivo RNA knockdown. So we keep asking why this anti-tag region will reduce the cleavage activities. Whether the anti-tag RNA can bond with the Cas13. So we use geofiltration to check different RNA substrates binding affinity with Cas13. We found different lengths of anti-tag could not block the binding with Cas13, and they can form stable complex with Cas13. So while they can block the cleavage activity of Cas13. So we determined the structures of the Cas13 in complex with anti-tag RNA. The anti-tag is highlighted here in red. So we found the anti-tag and tag form a stable RNA duplexes here. So in normal, before the RNA target load, the CRISPR-RNA repeater region, as I highlight here, they have a kink in -7 to -8 size. When it bonds with the target without the anti-tag region, they will form another kink here in between -1 to -3 region. Once it bonds with the anti-tag substrate, it will form a stable RNA duplex here, which is different to the target RNA without the anti-tag region. So the difference between here is the another kink formation and the RNA duplex. So why it will block the Cas13's activity? We then further analyze the structures. We found the extended RNA tag duplex will insert into the RNA binding channels as we show here, and will occupy the positions of the Cas13. When it bound with here is the pre-target bond of the Cas13 sequence. You can see the channel is narrow here. When it bound with the normal RNA targets without the anti-tag, it will located in the RNA binding channels, as we highlight here. Once it forms the extended the RNA duplex, the binding channel will move together to form a large channel to accommodate the complexes, and then will occupy the part of the binding channels and move here. Then once it's bound with the RNA duplex, the RNA Cas13 will locked into inactive state. The key residues is kept apart each other to lock it into an inactive site. Then we keep checking the whole complex. We found that the forming duplex will block the movements between the two happen domains and lock it into an inactive site. We further to mutate all these residues and all CRISPR RNAs to see whether it will reduce the activities as we show here. Also, we check the in vivo RNA interferences assay, and we found all these mutants will reduce the RNA interference activities. Later, we ask these discrimination mechanisms exist in other Cas13 systems. So we check the Cas13 we used, and we found that actually this extended base pairing between tag and anti-tag region will reduce the Cas13 activities. Take-home message actually is extended complementary between tag and anti-tag will lock the Cas13 in inactive states, and here will trigger the domain rearrangement of the Cas13. Okay, I'm gonna thank all the grants and all my lab members and all my colleagues, and thank you for attention.
Thanks a lot, Hui. So that's a very elegant study. So you are still welcome to put question in the Q&A, even though it doesn't have enough time. I will just ask a very shallow question. So for the case which targets DNA, they are like Cas9, the single protein one, and the Cascade system. But in contrast, for the Cas targeting RNA, it looks like very similar. It's like 1 or 2 proteins be able to do that job. What's the reason? Do you have any speculation of the reason or the design of these differences?
You mean between Cas13 and Cas9,
or maybe like there are for the Cas targeting DNA, there are multi-protein system and the single protein system, right? But
Yeah.
for the RNA, is there also a multi-protein system, or most of them are like a single protein system?
Actually, there are multiple systems for targeting RNA. Type 3 systems, it can be used in bacterial cells when manipulation of RNA. Because the reconstruction in mammalian cells is difficult for the multiple subunits, so it limits the applications.
I see. Okay, thanks a lot. So, Martina, do you want to close this seminar, or just I will just end up this seminar?
Yes, we can end it.
Okay. Okay.
Thank you, everyone.
Okay. So thanks everybody for joining. Thanks for your help. So thanks Hui and Rujuan give such a wonderful talk. Thank you. See you next time.
Thank you,
Thank
Paul.
you.
Thank you.